On the 26th of October 1873, two fishermen from Newfoundland named Theophilus Picot and Daniel Squires were rowing over the icy waters of Portugal Cove in Conception Bay. It was an ordinary morning of fishing, but in the distance, floating beneath the cliffs, they saw something that did not match any known marine debris. It was large, it was strange, and it was moving. When they approached with a gaff to investigate, the gelatinous mass reacted. It had a beak. It had tentacles. And before they could process what they were seeing, two enormous fleshy appendages emerged from that mass and began to coil around the boat. Picot took the axe he carried on board and severed both tentacles at once. The animal expelled gallons of black ink that darkened the water for hundreds of meters around and disappeared into the depths forever. Back in port, the two men brought with them one of the severed tentacles, which measured more than 5.5 meters in length and gave off an unbearable smell. The local rector, Moses Harvey, immediately recognized the importance of what he had before his eyes. It was the first physical and tangible proof of an animal that for centuries had been considered an invention of drunken sailors or superstitious minds. He photographed it, measured it, and sent a report to the Annals and Magazine of Natural History. The animal was a giant squid, an Architeuthis. The monster not only existed, but it also had a scientific name. That moment put an end to one of the fiercest debates of Victorian science. It was not a minor battle. Renowned scientists had ridiculed for decades sailors and ship captains who swore they had seen creatures of impossible proportions. In 1848, Captain Peter Macquarie of HMS Daedalus reported having sighted a creature more than 18 meters long near the Cape of Good Hope, describing the animal at such a short distance that, in his own words, if it had been a man, he would have distinguished its facial features. The paleontologist Richard Owen, the same man who coined the word dinosaur, not only dismissed the testimony, but publicly attacked the credibility of the British Royal Navy officer, insinuating in the press that the captain was either a liar or an imbecile, Macquarie insisted. The debate continued for years in scientific circles and in the press on both sides of the Atlantic. When Picot's tentacle appeared, all those who had bet on the existence of the monster were vindicated. The recovered tentacle was coiled like a snake, photographed and exhibited in the local museum of St. John's. Harvey, who was an amateur naturalist in addition to being a rector, knew what it meant to have a tangible specimen in hand. He presented it with all the solemnity that a discovery of the century deserves, and the British Scientific Journal published the set under the title Gigantic Cuttlefish in Newfoundland, an incorrect classification but understandable given the widespread astonishment of the time. In 1883, only a decade after the incident, the International Fisheries Exhibition in London displayed a scale replica of the giant squid suspended above the heads of visitors. The Victorian public, with their top hats and long skirts, looked up with a mixture of horror and fascination that no one in that room would have been able to fully explain. The story of the Kraken is, above all, the story of how human beings relate to what they cannot see or understand. The ocean covers more than 70% of the planet's surface. However, we know the topography of the moon in greater detail than the bottom of our own seas. In the mid-19th century, science still debated whether it was possible for life to exist at great depths. The pressure of the water, the absolute cold, the total darkness, it seemed inconceivable that anything could survive down there. Sailors knew something different. Whalers hunting sperm whales in the North Atlantic observed that these animals, when harpooned, would submerge for hours to enormous depths before returning to the surface. And when they killed them and opened them, they found in their stomachs horny beaks the size of human fists, circular scars on the skin that did not correspond to any known animal, and remains of tentacles of proportions that had no equivalent in the bestiaries of the time. Sperm whales were eating something enormous, something that lived very deep. The legends of the Kraken go much further back. Medieval Norwegian sailors described a creature so immense that it could be mistaken for an island. The animal would emerge. Sailors would disembark on what they believed was solid ground, and when the monster submerged, it would drag them with it. Pontoppidan, Bishop of Bergen in the 18th century, catalogued it with full seriousness in his Natural History of Norway as the largest of all animals in the world, a being capable of dragging entire ships into the abyss with its arms, and whose presence could be recognized because the schools of fish surrounding it were so dense that sailors could take them out of the water by hand. The Greeks had Scylla, the six-headed monster that devoured sailors in the Strait of Messina. Homer mentions it in the Odyssey. In Norse mythology, Jörmungandr, the world serpent that encircles the entire earth with its body, can be traced in part to those same traditions of colossal sea monsters. Jules Verne brought the creature to popular culture with 20,000 Leagues Under the Sea in 1870, where a giant squid attacks the Nautilus and devours a crew member. Even Alfred Tennyson dedicated a sonnet to it in 1,830 inches, which he described, "The old and sleepy Kraken, sleeping in the depths, far beneath the stormy waves." All these stories pointed, with greater or lesser distortion, toward something real. The monster was not a metaphor, it was an animal. What makes the story of the Kraken particularly notable is the tenacity with which popular narrative sustained for centuries the existence of something that official science denied with equal tenacity. Sailors did not have access to laboratories or scientific journals. They did not have the ability to formulate hypotheses systematically or to design experiments to test their claims. But they had something that armchair scientists did not: direct and accumulated experience of the ocean. They spent their lives at sea, in all possible conditions, and what they saw they described with the precision that their cultural references and available vocabulary allowed them. When they described a huge animal with tentacles attacking their vessels, they were not inventing, they were reporting, with the tools they had, something they had witnessed. The problem was the interpretation, not the observation. But the story of Architeuthis, the giant squid, begins about 542 million years ago. At that point in the planet's history, one of the most extraordinary events of evolution occurred, the Cambrian explosion. Before that moment, life existed on Earth, but it was mostly unicellular, slow, without defined form. Suddenly, in geological terms, the ocean filled with complex life. Eyes appeared. Jaws appeared. Structures for attacking and defending appeared. Among the first animals that arose in that evolutionary fervor were the mollusks. And within the mollusks, an extraordinary group that over time would give rise to all cephalopods: squids, octopuses, cuttlefish, and nautiluses. The oldest known ancestor of all these animals may be a small organism called Kimberella, which lived approximately 555 million years ago, even before the Cambrian explosion. It had a shell, a soft body, and probably a radula, that rasping tongue-like structure that characterizes almost all mollusks. If the classification of Kimberella as a mollusk proves correct, the evolutionary history of cephalopods will have to be rewritten to begin much earlier than previously thought, and the hypothetical common ancestor of cephalopods and vertebrates, which paleobiologists call Urbilateria, will also be older than currently estimated. From the Cambrian emerged the first cephalopod proper, an animal called Plectronoceras that crawled along the seafloor carrying an upright shell shaped like a cow's horn. Inside that protective shell were its vital organs. It was a primitive but effective predator. Over the following millions of years, cephalopods evolved toward increasingly specialized and sophisticated forms. In the Ordovician period, between 485 and 444 million years ago, cephalopods reigned over the oceans. Camaraceras, with its shell that could reach 9 meters in length, was possibly the apex predator of the seas of that period, in an ocean that covered almost the entire northern hemisphere of the planet. The seas of the Ordovician were warm and rich in life. It was, in a sense, the moment of greatest splendor of shelled cephalopods. But evolution does not stop. When the supercontinent Gondwana began to move toward the South Pole and the oceans cooled, a mass extinction wiped out much of the marine life of the period. Cephalopods survived. They survived the next period, and the next. Across 5 mass extinctions, the cephalopod lineage persisted and diversified. The great evolutionary bet of modern cephalopods was to get rid of the shell. The shell protects, but it also limits speed and maneuverability. Squids, octopuses, and cuttlefish traded the passive protection of a shell for speed, intelligence, and the ability to camouflage. The result was one of the most adaptable and sophisticated animal groups evolution has produced. Inside almost all modern squids persists a vestige of that ancestral shell, a transparent and flexible internal structure called a pen, made of a material similar to human nails, which functions as a pseudoskeleton to which muscles are anchored. In the cuttlefish, that vestige is the cuttlebone, the porous structure that bird owners know well because it is placed in cages as a source of calcium. In the octopus, almost nothing remains, only some traces in the tissue. Today there are approximately 1,000 species of cephalopods. They inhabit all the oceans of the planet except the Black Sea and the Baltic Sea. They range from the octopus, Octopus wolfi, which measures just over 1 centimeter and weighs a fraction of a gram, to the giant squid and the colossal squid, which are the largest invertebrates in the known world. The teuthologist James Wood once described them as the weeds of the ocean, resilient, adaptable, and capable of thriving in conditions that would eliminate most other animals. A defining characteristic of modern cephalopods as a group is their short and intense life cycle. Most species live between 1 and 2 years, reach sexual maturity quickly, reproduce massively, and die shortly afterward. This fast life strategy has important evolutionary implications. Generations follow one another rapidly, which means that natural selection can act more quickly than in long-lived animals. In a changing ocean, cephalopods can adapt in a few generations to conditions that would take decades to affect cetaceans or sharks. This evolutionary agility may be one of the reasons why the group has survived 5 mass extinctions while lineages of apparently greater complexity disappeared without leaving descendants. The Architeuthis ducks, the giant squid, is an animal that seems designed to challenge human intuitions about what a living being can be. An adult specimen can measure between 10 and 14 meters in total length, counting from the tip of the mantle to the ends of the feeding tentacles. The mantle alone, the part of the body that contains the vital organs, can exceed 2 meters. However, despite its enormous size, the animal lacks bones or any rigid skeletal structure. It is, fundamentally, a sack of muscle and water. The body of the giant squid has 3 essential parts. The mantle is the main mass, the functional equivalent of the torso in a vertebrate. It contains the stomach, the liver, the gills, the heart, and the reproductive organs. The head, clearly differentiated, houses the eyes and the buccal apparatus, dominated by a horny beak, similar in structure to that of a parrot, surprisingly small for the size of the animal. In most studied specimens, the beak fits in the palm of an adult hand. The 10 appendages, 8 arms, and 2 longer feeding tentacles surround that beak and carry food toward it once captured. The feeding tentacles are perhaps the most extraordinary feature of Architeuthis. At rest, they retract close to the body. When the animal detects prey, they extend with explosive speed. Images captured in 2004 and 2005 by the Japanese scientist Tsunemi Kubadera show how the giant squid approaches its prey with the feeding tentacles deployed, pointing directly at the target, and wraps them around it in a fraction of a second. At the ends of those tentacles are suckers surrounded by sharp, horny teeth that embed into the flesh of the prey to hold it. The elasticity of those tentacles in life can cause their length to vary considerably. South African expert Martina Roeleveld documented that the length of feeding tentacles in live specimens can range between 23 and 832% of the mantle length, which explains why size estimates of the animal based on measurements of dead specimens sometimes turn out to be so contradictory. In death, the tentacles lose their elasticity and are measured with values different from those they would have in life. The eyes of the giant squid are the largest in the animal kingdom in absolute terms. They can reach 30 centimeters in diameter, the size of a serving plate. This disproportion responds to a specific need. The animal lives in the mesopelagic and bathypelagic zones of the ocean, between 200 and 1,000 meters deep, where natural light is scarce or directly non-existent. Eyes of that size are capable of detecting the bioluminescence produced by other organisms in that darkness, which allows it both to hunt and to detect its predators, in particular sperm whales, which dive to those depths to actively search for it. The circulatory system of the giant squid is equally peculiar. The blood is blue because, instead of iron-based hemoglobin like ours, it uses hemocyanin, a protein that contains copper. Copper transports oxygen less efficiently than iron under conditions of high temperature and low pressure, but works remarkably well in the cold and deep waters where Architeuthis lives. The animal has 3 hearts, one central and 2 branchial, located next to the gills. This multiplication of hearts responds to the difficulty of circulating hemocyanin with sufficient efficiency to maintain the muscular activity of an animal of that size. An additional peculiarity explains why the flesh of the giant squid has a taste that scientists who have tried it, among them Clyde Roper of the Smithsonian Institution and Bill Gilley of Stanford, describe in terms ranging from ammonia to window-cleaning liquid or battery acid. The animal's tissues contain ammonium chloride in high concentration. Ammonium is less dense than salt water, which allows the animal to maintain almost neutral buoyancy without needing to swim actively. The same characteristic that gives it an energetically efficient ecological niche makes its flesh basically inedible for predators that try it for the first time, with the exception of the sperm whale, which swallows it without apparent difficulty and in enormous quantities. The debate about the behavior and temperament of the giant squid has accompanied the species since its first documented sightings. The account of Picot and Squires describes it as aggressive and dangerous. Other, more recent sources present a different picture. The only deep-sea footage of a live Architeuthis obtained by Kubodera shows an animal focused on hunting but not particularly bellicose in the sense of attacking objects that are not prey. When a specimen appears at the surface, something that happens rarely, scientists agree that it is most likely gravely ill or dying. Roper, who devoted decades of his career to the search for the living giant squid, stated on several occasions that he would like to swim alongside one if he had the opportunity. His more cautious colleagues point out that such enthusiasm may not sufficiently take into account the destructive capacity of the animal's toothed suckers. The honest answer is that no one knows for certain. The giant squid has never been observed in its natural environment for long enough to characterize its behavior rigorously. What is clear is that the animal Picot encountered at the surface was probably dying, disoriented, out of its habitat, and reacted in the only way an animal in those circumstances can react. It was not an ambush. It was panic. Until the end of the 20th century, Architeuthis was considered the largest invertebrate on the planet. That certainty began to crack when scientists started to examine more closely the stomach contents of sperm whales captured in the Southern Ocean, the sea that surrounds Antarctica. They found chitinous beaks that were significantly larger than any Architeuthis beak they had ever seen, too large. The animal to which those beaks belonged was given the scientific name Mesonicoteuthis hamiltoni, the colossal squid, and was formally described for the first time in 1925 from fragments recovered from the stomach of a sperm whale. For decades, its existence was almost a footnote in zoology. That changed drastically in 2007 when New Zealand fishermen operating in Antarctic waters hauled onto the deck a live specimen of colossal squid weighing more than 450 kilograms. It had a mantle more than 2.5 meters long. It was the largest invertebrate ever captured alive. Architeuthis surpasses Architeuthis in body mass decisively. Its mantle is proportionally much more robust and muscular, but perhaps the most striking difference lies in the tentacles. While the giant squid has toothed suckers, the colossal squid possesses rotating hooks at the ends of the feeding tentacles, structures that function as rotating barbs capable of embedding into the flesh of the prey with a force difficult to counteract. Any prey that gets caught in those hooks and tries to free itself by rotating in the opposite direction only causes the hooks to dig in further. The eyes of the colossal squid are also the largest known in absolute terms. They can exceed 30 centimeters in diameter and are equipped with bioluminescent photophores on the rear side, a kind of headlights that project light forward in the darkness of the depths to illuminate the target before the attack. Analyses of the chemistry of the beaks of both species, carried out by scientists Yves Charel and Keith Hobson, revealed that the colossal squid feeds at higher trophic levels than the giant squid. Architeuthis does not hunt medium-sized squid and fish. It competes directly at the feeding level of large sharks and toothed cetaceans. This suggests that, in the depths of the Southern Ocean, the colossal squid is a top-tier superpredator, not a potential prey but an active rival of sperm whales that also hunt in those waters. Hook marks from Architeuthis have been found on the skin of sperm whales. The interaction between these two colossi of the depths is not fully resolved and probably includes episodes of predation in both directions, depending on the relative size of the individuals involved. What we know about the colossal squid remains fragmentary. Most recovered specimens have arrived dead or dying. Its life habits, its reproductive patterns, its social behavior, if any exists, are largely unknown. The number of individuals in the ocean is an unknown. What analyses of beaks found in sperm whales do suggest is that the population may be considerably larger than its apparent rarity would indicate. The problem is not that Architeuthis is scarce. The problem is that it lives in the most inaccessible place on the planet, the depths of the Southern Ocean, surrounded by ice and storms for most of the year. However, there is a paradox at the bottom of the ocean. It is the darkest place on the planet, absolutely deprived of sunlight beyond 600 meters deep, and yet it is full of light. Approximately 90% of the organisms that live in the ocean depths produce or emit some form of bioluminescence. The seafloor is not black. It is a universe of flashes, pulses, and coded light signals that animals use to hunt, hide, communicate, and reproduce. Aristotle described this luminescence in his writings 2,500 years ago. What he could not know is what it would eventually contribute 20 centuries later to one of the most important advances in modern neuroscience. Cephalopods are absolute masters of light and color. Even shallow water squids have in their skin an architecture of chromatic cells of a complexity that has no precedent in the animal kingdom. There are 3 cellular layers involved. The outermost contains chromatophores, cells that carry yellow, red, black, or brown pigments enclosed in elastic sacs. Each sac is connected to between 4 and 24 muscle cells. When those muscles contract, the sac expands up to 7 times its resting diameter and the color becomes visible. When the muscles relax, the color disappears. This can happen dozens of times per second, producing the rippling and flashing patterns that characterize cephalopod visual communication. Beneath the chromatophores are iridophores, cells that do not produce color but reflect light like metallic surfaces, generating blues, greens, and golds. They do not open or close, they simply redirect light. They are responsible for the iridescent flashes seen on the skin of many cephalopods. In the deepest layer are the leucophores, flat cells that passively reflect the color of the animal's immediate surroundings, completing the camouflage system. The result of this three-layer cellular architecture is the ability to produce visual patterns in real time of a complexity that finds no equivalent in any other animal. Australian researcher Mark Norman described cuttlefish skin as covered with television screens. The metaphor is precise. The animal can make color patterns ripple across its body, create optical illusions of movement, mimic the texture and chromatic palette of any surface in its environment, and all this in milliseconds. The laboratory of Roger Hanlon at the Marine Biological Laboratory in Woods Hole has recorded on video cuttlefish that reproduce the texture of black and white checkerboards, of gravel beds of different sizes, and of surfaces they have never encountered in their natural environment. This suggests that the camouflage system is not a fixed set of pre-programmed responses, but a process of active visual computation that generates adaptive patterns in real time. The United States Department of Defense has funded research on this system in the hope of transferring its principles to the design of camouflage for soldiers. The cuttlefish takes this art to the extreme because, unlike squids, it cannot flee into the depths to escape predators. The cuttlebone is destroyed at great pressures. Therefore, the cuttlefish has developed the most sophisticated camouflage system of all cephalopods. Its survival depends on becoming invisible rather than escaping. Australian dolphins, for their part, have developed specific techniques to dismember cuttlefish and remove both the cuttlebone and the ink before eating them, behaviour that has been documented in the Spencer Gulf of South Australia and is transmitted culturally from one generation to the next. There is a profound irony in all this. Despite the extraordinary elaboration of the chromatic system of cephalopods, these animals are almost certainly colorblind. Their eyes have a single type of photoreceptor, which in principle prevents them from distinguishing colors as we do. Scientists have proposed that they might use differences in the polarization of light to discriminate between wavelengths, which would constitute an alternative mechanism for color perception. Whatever the exact mechanism, the result is that the most sophisticated animal in the world, in terms of color production, probably cannot see the spectacle it itself generates. It produces colors because other animals see them, not because it sees them. In the depths, where sunlight does not penetrate, cephalopods have developed bioluminescent strategies of astonishing diversity. The Taningia danae, a deep-sea squid that lacks feeding tentacles but has exceptionally long arms, possesses the largest photophores known in the animal kingdom, located at the ends of two of its arms. When it attacks, those photophores emit bright flashes just before impact. Scientists believe the function may be twofold: to disorient prey and to more precisely calibrate the distance to the target. The Hawaiian bobtail squid has developed one of the most ingenious adaptations to manage bioluminescence. It does not produce light directly. It houses colonies of the bacterium Vibrio fischeri in a specialized organ in its mantle. Each dawn, it expels most of those bacteria. During the day, it rests buried in the sand. When night falls and it goes out to hunt in shallow waters, illuminated by the moon and stars, the remaining bacteria have multiplied again to reach the density required to emit light. That light, projected from the squid's underside downward, mimics the glow of the water's surface seen from below, making the animal invisible to predators swimming beneath it. Researcher Margaret McFall-Nye has demonstrated that the relationship between the bobtail and its bacteria is not simple coexistence. It is an intimate association in which each part modifies the other. When the first Vibrio fischeri bacteria colonize the light organ of the newly hatched squid, the bacteria trigger changes in the development of the organ's tissue that do not occur in their absence. The organ only completes its maturation when the bacteria are present. The bacteria and the squid need each other in the most literal sense. This system has proven to be an extraordinarily useful research model for understanding how mammals, including humans, develop their intestinal microbiome. Human babies born by caesarean section do not pass through the birth canal and do not receive the first dose of beneficial bacteria at the moment evolution has anticipated, in a similar way to how a bobtail squid that grew without bacteria would never complete the development of its light organ. The brain of cephalopods is an evolutionary anomaly. In vertebrates, including humans, the nervous system is highly centralized. The brain controls the rest of the body through the spinal cord. Cephalopods also have a central brain, but approximately 3/5 of their nervous system resides outside it, in the arms and tentacles. Each arm of an octopus has its own nerve ganglion and is capable of making decisions and executing actions semi-independently. If an arm is severed from the body, it can continue functioning for hours, responding to environmental stimuli with apparent purpose. This decentralized architecture has profound consequences for cognition. An octopus does not give instructions to each arm individually. It gives a general command, and the arms interpret it locally. When an octopus inserts an arm into a crevice to explore whether there is food, the arm processes tactile and chemical information autonomously. The brain receives only a summary. This system is incredibly efficient for animals that need to coordinate 8 independent limbs with minimal latency. Each sucker of an octopus is, in turn, capable of semi-independent operations. They can pass an object from one sucker to the next along the entire arm, like a conveyor belt of flesh. The ratio between brain weight and body weight in cephalopods places them above fish and reptiles and below most birds and mammals. This metric is not infallible as an indicator of intelligence, but combined with observations of behavior in captivity and in the wild, it suggests remarkable cognitive capacities. Octopuses have been observed using tools. They collect coconut shells on the seafloor and transport them, awkwardly walking on their own arms in what scientists have called stilt walking, to use them later as shelter. They solve mazes, learn to open screw-top jars, recognize individual caretakers in aquariums, and exhibit differentiated behaviors depending on the human individual approaching their tank. Cuttlefish take communicative complexity further. They use their skin as an active communication channel, emitting visual patterns that researchers have interpreted as courtship signals, warnings to rivals, and status messages. Scientist Roger Hanlon has documented how male cuttlefish can simultaneously display courtship patterns on the side of the body facing females and appeasement patterns toward other males on the opposite side, a capacity for parallel communication in 2 independent visual channels. No other known animal does anything functionally equivalent. Humboldt squid, Dosidicus gigas, have been observed hunting in coordinated groups, reminiscent of the hunting tactics of dolphins or wolves. They use bioluminescent flashes and color changes during the hunt to coordinate. Biologist Bill Gilley of Stanford University described having observed hundreds of individuals maneuver in perfect synchrony, turning together in the darkness with a precision that suggested real-time communication. The Humboldt can also be considerably dangerous to humans. Mexican fishermen call it Diablo Rojo, partly because of the color it adopts when agitated, and partly because of stories of people who fell into the water among schools of Humboldts and reached shore with sucker marks on their bodies. However, other researchers, such as Gilley himself, have swum among them without special protection, without suffering any harm. The difference may lie in whether the animal is in a state of active hunting or not. Neuroscience has a colossal debt to cephalopods, and it owes it in a very specific way. For decades, the main obstacle to studying how neurons function was that human nerve cells are too small to be manipulated with the available tools. Human neurons have axons of a fraction of a millimeter in diameter. To study the electrical mechanisms of nerve signaling, scientists needed larger axons. The common squid Loligopeli has a giant axon that is visible to the naked eye. It can reach 1 millimeter in diameter, about 1,000 times thicker than a human axon. This axon exists because the squid needs to escape predators as quickly as possible. When the animal detects danger, it needs to simultaneously contract all the muscles of the mantle to expel water and propel itself backward in the shortest possible time. A very thick axon conducts electrical impulses more rapidly than a thin one. The evolutionary result was this nerve of extraordinary proportions that ended up being one of the most valuable instruments of 20th century neurobiology. In 1936, biologist John Young identified this giant axon in the laboratory at Woods Hole, Massachusetts. A decade later, Alan Hodgkin and Andrew Huxley inserted microelectrodes into the interior of that axon and measured for the first time with precision what happens electrically inside a nerve cell when it generates an impulse. They discovered that the process was based on the movement of sodium and potassium ions through specific channels in the cell membrane, opening and closing like microscopic gates in a precise sequence. This information revolutionized neuroscience. Hodgkin and Huxley received the Nobel Prize in Physiology in 1963. The informal remark of a colleague summarizes well the magnitude of the discovery. The Nobel Prize should be given to the squid. The later work of Clay Armstrong on the ion channels of the squid axon opened an entire branch of pharmacology. Calcium channel blockers, which are now routinely prescribed to treat high blood pressure and prevent heart attacks, are a direct consequence of that work. Some medications for epilepsy and for type 2 diabetes act on similar ion channels. An animal that lives hundreds of meters deep in the Pacific contributed decisively to the most common pharmacological treatments of contemporary Western medicine. At the same time, the bioluminescence of a jellyfish was the basis of the green fluorescent protein that allowed neuroscientists to illuminate individual cells within the brain without destroying them, opening the way to the study of Alzheimer's, Parkinson's, and amyotrophic lateral sclerosis at the molecular level. The greatest irony of this entire chain of discoveries is its completely accidental nature. No one designed research on the squid's giant axon with the intention of developing a drug for hypertension. No one studied bioluminescent jellyfish thinking about Alzheimer's. Basic scientific research, motivated by curiosity about how nature works, continuously produces knowledge that becomes useful decades later in ways that no one could have foreseen. The squid was studied because it was interesting, not because it was useful. Utility arrived on its own. This pattern repeats itself so many times in the history of science that it should be an irrefutable argument in favor of funding basic research on organisms that have no obvious immediate commercial application, including deep-sea cephalopods, whose biology we are only beginning to understand. For more than 100 years after the encounter of Theophilus picot, the giant squid was known exclusively from dead or dying specimens stranded on beaches, fragments found in the stomachs of sperm whales, or bodies recovered floating on the surface. No one had ever seen a living Architeuthis in its natural environment. This absence of direct observation fueled decades of speculation about the behavior, distribution, and biology of the animal. Clyde Roper, of the Smithsonian Institution, devoted more than 40 years to trying to solve that problem. His logical strategy was to follow sperm whales. If sperm whales eat giant squid, where there are sperm whales, there should be giant squid. He estimated that a sperm whale can consume between 50 and 100 Architeuthis per year, which implied, doing the math, that the total population of giant squid in the ocean must be enormous. There are many down there, he stated in several interviews, but we do not see them because we do not live where they live. In 1996, together with Greg Marshall of National Geographic, he approached female sperm whales in inflatable kayaks in the waters of the Azores and managed to attach submersible cameras to the heads of 2 of them. The images showed deep-sea marine life of great interest, no giant squid, in 1997, in the waters of New Zealand. Nothing. In 1999, in the Kaikoura Canyon, nothing. Success finally came in 2004 and 2005, but it was the Japanese Tsunami Kubodera who achieved it. Working with the same principle of using sperm whales as a guide, he lowered a baited fishing line to 900 meters deep in a submarine canyon off the Ogasawara Islands in southern Japan and placed a wide-angle camera. On the 30th of September 2004, that camera captured the first images of a living Architeuthis. The animal approached the bait with its feeding tentacles extended, launched them with explosive speed, and became hooked on the line. It struggled for 4 hours and 45 minutes before freeing one of its tentacles and escaping. The tentacle was recovered. It measured nearly 6 meters and was still contracting on the deck of the boat, with the suckers gripping any surface they touched. In 2006, Kubodera returned with better cameras and managed to film on video an Architeuthis in its natural environment, an animal between 4 and 5 meters in length, actively hunting in the darkness at 900 meters depth. The images showed that the giant squid approached prey with the 2 long feeding tentacles pointing forward in parallel, and wrapped them around it in a way that Kubodera and his co-author Kyoichi Mori described in the journal Proceedings of the Royal Society as similar to the way a python coils around its prey immediately after the attack. In 2012, researcher Edith Widder and an international team managed to film the first Architeuthis in the Gulf of Mexico using a different technique. Instead of conventional bait, they used a camera equipped with artificial bioluminescent light that imitated the luminous signals of organisms in distress. The giant squid was attracted by that signal, not by food, which suggests that giant squid may use the bioluminescence of their prey as an indicator of vulnerability before attacking. This footage showed an animal of intense reddish coloration, with its arms extended in a position of active exploration that was at once more graceful and more cautious than historical descriptions had suggested. One of the most persistent mysteries about the giant squid is how many species actually exist. Since the 19th century, more than 20 different species names have been proposed for Architeuthis. Most specialists believe that the real number is much lower. The most widespread opinion among teuthologists is that there are probably 3 distinct species, one in the North Atlantic, one in the North Pacific, and one in the Southern Ocean around Antarctica. However, a DNA analysis published in 2013, based on tissues from specimens around the world, found surprisingly low genetic diversity, which suggests that there could be a single global species with populations connected through larval dispersal by ocean currents. The debate continues. What is established is that Architeuthis is not a rare animal in terms of absolute population. The mathematics of consumption by sperm whales suggest that the population of Architeuthis could be enormous. What happens is that they live in the only place on the planet to which humans have extremely limited access, the ocean depths between 200 and 1,000 meters. In that habitat, they are practically invisible to us. Every time we find one, it is because it has come to the surface, which generally means that something has gone terribly wrong for the animal. In 2008, marine biologist Sean van Sommeren found the remains of an Architeuthis floating in Monterey Bay, California. It was only the 5th specimen ever recorded on the Californian coast. He brought it ashore, and it was dissected by a team of scientists from the National Marine Fisheries Service and Stanford University. Julie Stewart, then a doctoral candidate in Bill Gillies' laboratory, extracted the gills to compare them with those of the Humboldt squid. That was her primary research subject. Other researchers recovered the statoliths, small calcareous structures of the animal's vestibular system, similar to fish otoliths. Statoliths grow layer by layer during the life of the animal, like the rings of a tree, and can be used to estimate the age of the individual. The analysis of those statoliths had not been published at the time Wendy Williams's book on the Kraken went to print, and that perfectly illustrates a fundamental problem of Architeuthis biology. Data are scarce because specimens are scarce. It is debated whether the animal lives between 1 and 3 years, or if it can reach 12. It is not known whether males and females live in different areas of the ocean. It is not known whether there is any form of social behavior, or if they are completely solitary. It is not known whether they migrate vertically between shallow and deep waters, as many other squids do. It is not known exactly how they reproduce or where they lay their eggs. A school of giant squids, or a clutch of their eggs, has never been found. The toxicology of the Monterey specimen revealed something surprising. The levels of mercury and other contaminants in the tissues of the giant squid were very low, much lower than expected for a predator of its size. Sperm whales, bluefin tuna, or deep-water sharks accumulate contaminants along the entire trophic chain. The relative toxicological cleanliness of Architeuthis suggests that the animal spends very little time in contaminated coastal waters and that its diet includes prey of relatively low trophic levels, at least compared to what might be expected of a predator of that size. This reinforces the hypothesis that the giant squid may be an ambush predator rather than an active hunter. The animal uses ammonium chloride for neutral buoyancy, which means it does not have to swim actively to remain at its preferred depth. It can float almost effortlessly for prolonged periods. This characteristic, combined with the low levels of contaminants in its tissues, leads some researchers to propose that Architeuthis spends much of its life in a kind of active suspension, waiting in the darkness for suitable prey to pass within reach of its tentacles before executing the attack. It is a very different strategy from that of the Humboldt, an active group hunter, but perfectly compatible with the available data. The question of the specific intelligence of the giant squid is one of the most difficult to answer. Analysis of the brain of the Monterey specimen showed that the ganglia of Architeuthis are proportionally smaller than those of the Humboldt. This could indicate lower cognitive sophistication, or it could simply reflect a different lifestyle. An active group hunter like the Humboldt needs to process more social information than a solitary ambush hunter like Architeuthis. An octopus that lives in shallow waters among rocks and acts as prey for multiple predators has developed a tactical intelligence that a resident of the abyssal depths likely does not need. Intelligence, in evolution, is not a good that accumulates by accumulation. It is a specific response to specific environmental pressures. In 2015, the oceanographer Michael Vecchione and his colleagues documented for the first time in ROV images, remotely operated vehicles, a completely white and translucent octopus at more than 4 kilometers of depth in the Pacific. It was an animal of the Dumbo genus, with ear-shaped fins that it used to move slowly over the ocean floor. It was probably a new species. The formal description took several more years. This occurs regularly with deep-sea species. They are discovered in ROV images long before there is a physical specimen available for official scientific description. Current estimates suggest that between 70 and 95% of the species that live in the deep ocean are unknown to science. The deepest ocean trenches, such as the Mariana Trench, with its 11 kilometers of depth, have been visited by human beings on very few occasions and explored systematically to a much lesser extent. At those extreme depths, there are organisms adapted to pressures that would crush any vehicle not specifically designed to withstand them. What lives in those zones is, in practice, completely unknown. The possibility that cephalopods exist at those depths cannot be ruled out. Life forms that have survived 500 million years of evolutionary changes have demonstrated an adaptive capacity that should make us cautious before declaring any combination of environment and organism impossible. But really, the Kraken is above all a symbol that has functioned in very different ways in different cultures and times. In the seas of northern Europe, it was the ultimate threat of the ocean, the representation of everything that could destroy a vessel without leaving a trace. In 19th century literature, It became a metaphor for the unknown that lurks beneath the rational surface of the civilized world. Jules Verne used it to dramatize the possibilities of underwater exploration. Herman Melville evoked in Moby Dick a vast pulpy mass, furlongs in length and breadth, coiled and writhing like a nest of anacondas, that his narrator Ishmael describes as one of the most overwhelming phenomena that the secret seas have revealed to the human race. Alfred, Lord Tennyson, dedicated to it in 1831 of his first sonnets, describing the Kraken sleeping in the depths in an ancient and indolent sleep, surrounded by smaller monsters, waiting for an awakening that would only come at the end of time. The persistence of the Kraken as a cultural figure responds to something concrete. The deep ocean is the only truly inaccessible place on the planet for human beings. The moon is more mapped than the bottom of the Atlantic. Outer space, with all its incomprehensible emptiness, seems more familiar to us than the trenches of the Pacific because we can observe it with telescopes. The ocean floor requires special machinery to explore it. The pressure is lethal without protection, and the darkness is total. The monster that lives down there is the projection of that inaccessibility. When something cannot be seen, imagination fills it with the largest and most terrifying thing it can conceive. The difference between the 18th century and the 21st century is not that the monster has disappeared, it is that now we know what it is. The Architeuthis exists. The Architeuthis exists. And there are probably cephalopods in the ocean depths that have not yet been catalogued. Marine biology has repeatedly shown that the deep ocean holds surprises of great magnitude. The coelacanth, considered extinct for 65 million years, was rediscovered alive in 1938 off the coast of South Africa. The ghost shark Chimera, a primitive relative of sharks, was filmed for the first time on video in its natural environment in 2016 at a depth of 2.5 kilometers in the Atlantic, even though the species had been known since the 18th century from stranded specimens. There is something that all researchers who work with cephalopods mention with some version of the same words. When a squid or an octopus stares at you, there is something in that eye, a presence, that is difficult to ignore. The teuthologist Clyde Roper put it this way: when you look into their eyes, you know there is something there. It is not cheap anthropomorphism. It is the recognition that the eye of the cephalopod and the eye of the vertebrate arrived at structurally very similar solutions through completely separate evolutionary lineages over 500 million years. The squid's eye has a pupil, an iris, a lens, and a retina. It does not have a blind spot, unlike the human eye. It can perceive the polarization of light. It is, in several technical aspects, more capable than ours. The film industry and popular culture have kept the Kraken alive with uneven results. In Pirates of the Caribbean, the Kraken is a machine of mass destruction in the service of Davy Jones. In the television series based on the works of Lovecraft, it becomes a metaphor for incomprehensible cosmic forces. In dozens of marine horror novels, the giant squid is the recurring antagonist of the deep ocean. None of these representations has much to do with the real animal, but all share something with medieval legends. The use of the cephalopod as a vessel for fear of what cannot be seen. What science has done with the Kraken is not to demystify it in the sense of emptying it of interest. It is, in fact, the opposite. The real Architeuthis is more complex, stranger, and more fascinating than any fictional version. An animal that can hunt with tentacles that extend explosively, whose eyes are the size of a dinner plate, whose blood is blue, whose tissues smell like floor cleaner, that lives in absolute darkness at hundreds of meters of depth, and that we have only been able to observe 2 or 3 times in the entire history of zoology, does not need to be exaggerated to be extraordinary. There is also a philosophical dimension in the story of the Kraken that goes beyond zoology. The 19th century scientific battle between skeptics like Owen and believers like Harvey was not only a disagreement about an animal. It was a confrontation between two ways of relating to knowledge, the one that requires tangible evidence before admitting the unknown, and the one that accepts that the accumulated testimony of people who have been in places where scientists have not been may contain real information. The Victorian sailors who described the giant squid were not ignorant, nor were they inventing stories. They had access to a world that armchair scientists could not observe directly. The problem was not the credibility of the witnesses, but the lack of instruments to verify their accounts. When the instruments arrived, in the form of severed tentacles and stranded specimens, the witnesses were vindicated. That lesson has broader applications. The deep ocean is still full of things that sailors, fishermen, and divers describe and that science has not been able to confirm or rule out. Not all of those descriptions point to real animals. But the precedent of Architeuthis suggests that epistemological caution goes in both directions, not only in the face of what seems impossible, but also in the face of the systematic dismissal of testimony from people who spend their lives in direct contact with the sea. The inventory of what is known about Architeuthis in 2025 is substantially greater than that of 1,873, but it remains fragmentary. The existence of at least one and possibly 3 species has been confirmed. It is known that adults live between 200 and 1,000 meters of depth in mesopelagic zones of the Atlantic, Pacific, and Indian Oceans. DNA analyses of tissue samples suggest a surprisingly low genetic diversity for an animal with such a wide distribution, which points to a genetically homogeneous population on a global scale. The maximum documented size of fully recovered specimens is just over 13 meters in total length, although the beaks found in sperm whales suggest the existence of considerably larger individuals. It is known that sperm whales are the main predator of the giant squid, and that a sperm whale can consume several Architeuthis per week. It is known that the tissue of the giant squid contains high levels of ammonium chloride that give it neutral buoyancy. It is known that the flesh has an unpleasant taste for humans. It is known that the animal has 3 hearts, blue blood, the largest eyes in the animal kingdom, and a chitinous beak that is organically one of the hardest materials produced by nature. It is known that its feeding tentacles can function semi-autonomously for hours after the death of the animal. And it is known that Architeuthis is, in all probability, an animal much more common in the deep ocean than its apparent rarity suggests. What is not known includes the exact longevity of the animal, the patterns of hunting and feeding behavior in situ beyond what has been observed in the few available recordings, whether there is any form of social structure or communication between individuals, how they reproduce and where they lay their eggs, whether they migrate between different depths or latitudes. What happens to the bodies when the animal dies in the depths, and how many individuals exist in total in the world ocean? The colossal squid raises even more fundamental questions. Even its maximum size range remains uncertain. Several shallow-water cephalopods are changing their geographic distribution ranges in ways that coincide with ocean warming. The Humboldt squid, historically confined to tropical and subtropical seas of the eastern Pacific, has extended its presence northward as far as Alaska in recent decades. Massive strandings of Humboldts on California beaches have become more frequent. On the east coast of the United States, squids of the species Architeuthis plae, which historically wintered in warm waters south of Cape Hatteras, are changing their migration patterns. These variations have implications for coastal ecosystems and for fisheries, but they are still not sufficiently studied to draw definitive conclusions. Research on cephalopods faces a structural funding problem that other fields do not have. There is no large-scale fishing industry that depends on Architeuthis. There is no equivalent of tuna or salmon that generates economic pressure to finance its study. Knowledge of the giant squid advances mainly thanks to opportunistic findings, such as that of Van Sommeren in Monterey, to sperm whale research expeditions that accidentally come across specimens, and to the work of a handful of dedicated teuthologists who work with budgets that most vertebrate biologists would find scarce. The conservation status of Architeuthis is another unknown. Without knowing how many individuals exist, it is impossible to assess whether the species is threatened or not. Changes in the deep ocean produced by global warming, ocean acidification, and plastic pollution potentially affect the habitat of the giant squid, but in ways that we cannot quantify because we do not know enough about its biology. It is possible that giant squids are as resilient to environmental changes as the rest of the cephalopod lineage has proven to be over 500 million years of extinctions and recoveries. It is also possible that they are more vulnerable than we think. Without data, there is no answer. There is a connection between the different discoveries that cephalopods have made possible that goes beyond zoology. The squid neuron, the bobtail photophore, the cuttlefish chromatophore, and the blue-ringed octopus venom are different evolutionary responses to fundamental problems of biology: how to send signals, how to produce light, how to change appearance, how to kill or immobilize prey. Each of these systems was refined over hundreds of millions of years in a direction that vertebrate evolution did not take, or took differently. When researchers study these systems, they find solutions that vertebrate biochemistry had not explored. Those alternative solutions are the true scientific value of cephalopods. The neuroscientist Vincent Peerybone from Yale University, who spent years studying the green fluorescent protein of jellyfish and its applications in neuroscience, put it in a way that is worth remembering. We have libraries of solutions that we can capture from other animals. It is extraordinary to learn how strange the world under the ocean is. Those animals can provide us with fantastic tools that do not exist in our own world. The idea that the deep ocean is a library of unexplored biological solutions is possibly the most concrete reason to protect and study it. In the end, the Kraken was real. What was not real was the image that had been constructed of it, an active and destructive monster that waited in the depths to devastate ships. Architeuthis is in reality an animal that spends most of its existence in the darkness of the ocean depths, probably floating almost motionless in the water column, waiting for prey to pass within reach of its tentacles, being in turn hunted by sperm whales that dive for hours to find it. The difference between the mythical monster and the real animal says something about the mechanisms of human imagination in the face of the unknown. The void of information is filled with what is most terrifying possible. A large and incomprehensible animal that lives where we cannot see it automatically becomes a threat. When we finally have the real data, what we find is invariably more complex and more interesting than the monster we imagined. In 2009, when the marine biologist Sean Van Sommeren found the remains of Architeuthis in Monterey Bay, the specimen was dissected in a laboratory surrounded by scientists and television cameras, and the tissues were sent to multiple institutions for analysis. The same creature that had terrified medieval sailors, that had inspired Verne and Melville, that had been denied and then affirmed by Victorian science, was being methodically divided into freezer samples. It was not a demystification, it was a change of perspective. The giant squid did not become less extraordinary by being known. It became more extraordinary. It turned out to be an animal with the largest eyes in nature, with blue blood, with tentacles capable of continuing to respond to stimuli after death, with tissues that had absorbed so few contaminants that they were puzzling to toxicologists, with a body architecture that has no equivalent in any other animal of its size. And behind it, in even deeper and colder waters, a relative even larger and more formidable, the colossal squid, of which we know even less. The deep ocean continues to keep its secrets. What is known is that it is not an empty and hostile place. It is the largest ecosystem on Earth, and it is full of life that produces light, that changes color, that communicates in frequencies that human beings cannot directly perceive, that has solved the problems of survival in ways that continue to surprise biologists. The Kraken that emerged from the depths into mythology was a reflection of the fear of the unknown. The Architeuthis that emerges into science is an argument in favor of continuing to look downward. The tools available to explore the deep ocean improve every decade. Remotely operated vehicles can now descend to depths that would have been impossible 20 years ago and transmit high-definition images in real time. Autonomous submersible cameras can be programmed to remain in one place for hours or days, waiting for whatever passes in front of their lenses. Acoustic sensors can detect the presence of large animals in the water column without needing to see them directly. And environmental DNA techniques, which consist of analyzing water samples in search of genetic material from animals that have passed through that area, are beginning to be applied to the study of deep-sea cephalopods. With these tools, the coming decades should produce more knowledge about Architeuthis than all previous centuries combined. The monster of the abyss still has much to teach. Meanwhile, the Kraken remains in the depths. That fact in itself, the persistence of the unknown about a 14-meter animal that inhabits the largest ecosystem on the planet, is a reminder of the scale of what remains to be explored. Human beings have placed a vehicle on Mars, have photographed black holes millions of light years away, and have deciphered the genome of thousands of species. And yet, they still do not know where Architeuthis lays its eggs, how long it lives, or how many individuals are swimming right now in the ocean depths. That asymmetry between what we know about the distant cosmos and what we ignore about the nearby ocean is one of the great scientific facts of our time.